For today, Professor and Director of the Institute of Neuropathology at the University of Zurich. Professor Aguzzi has led groundbreaking molecular studies on the basic biology of neurological diseases caused by prions. His work has been instrumental in our understanding of how prions enter the body and travel to the brain to trigger neurological disease. And in addition, his work has contributed to the understanding of other diseases involving aberrantly folded proteins, such as Alzheimer's disease. So among other honors, Professor Aguzzi has won the Ernst Young Prize, the Robert Koch Prize, l'Artois Ballier Latour Health Prize, and the Medal of European Molecular Biology Organization. He also has held 2 ERC advanced grants. Professor Aguzzi's work is a great example of how persistent curiosity-based research on one single protein can lead to repeated breakthroughs over many years. This is also reflected in his title, 30 Years on a Single Protein, the Do's and Don'ts in Prion Research and Other Curiosity-Driven Endeavors. Excuse me. Welcome, Professor Aguzzi.
Thank you, Rosalind. So great to be here, and I am very connected to your university since many years, and I've been serving on your advisory board now for several years, and it has always been a pleasure. I have immensely enjoyed the interdisciplinarity that you have been able to create within your departments, and also several of you I have been collaborating scientifically also for many years. Walker Jackson, who is in the audience, is a great friend since a long time, and also a collaborator, and whom I knew since his days at the Whitehead Institute, but more importantly, really, one of your colleagues is really one of the person who has changed my scientific life forever, and that was really Peter Nilsson, who came to my lab as a postdoc. I mean, we are talking like 15 years ago. And Peter, and I tell you the story because it was really amazing. Out of the blue sky, Peter sends me an email, said, "Look, I am a chemist, and I did my PhD on this filamentous thiophene compounds, and I have this idea that they could actually interact with amyloids, and can I come to your lab and do work on it?" I said, "Yeah, sure, come tomorrow." Perhaps I should say, well, the rest was history, because this really completely changed my view on how to detect protein aggregates, order protein aggregates of like prions and Alzheimer plaques, and has spawned an area of research that we are still very active on. And in fact, I will talk about our new results on this. So it was really a complete change of direction for my lab, but also, and I think that this is something that we may discuss also then afterwards in the session with the young scientists. And I think that it was a great example of interdisciplinarity, and because with Peter as essentially an organic chemist, you couldn't go further away from what we were doing. Well, maybe yes, maybe I could have hired somebody, an expert in medieval art, but what Peter was doing was already pretty far away from our expertise, and this catalyzed so many different activities, and so we started on very simple things, and on detecting prions, and then detecting prion strains, and then went on to Alzheimer's, and then first for several years nothing worked, but then all of a sudden things that did start to work, and then it became again very exciting, and now essentially the inventions and the discoveries that Peter has made while he was with us, I mean, this is being used in a lot of labs worldwide. I mean, it has become kind of a standard procedure. And same thing with Per Hammerstrom, who has been incredibly generous and cooperative, and with whom we have made a lot of things. So I have to say, by and large, the experience with Linköping University has been really a complete, has changed my life. And right, so let me see if I can share a couple of slides with you. And so I will have to click a couple of things now. And right, here we go. So the I have to say that I had given a pretty generalistic title to David and Rosalind originally, because it was not entirely clear to me what the audience was. Now, after talking to David, it has become clear, and I think that I would, what I'm going to do in the next 30 minutes or so is to expose you to some of the recent research of the lab, and particularly the work that has been going on the polythiophene and how this is now developing, but I will talk a lot about methods, and the reason is that I hope that some of these methods might be useful to you. And so one thing that I was really keen to do was to use fluorescence to track prions, how they spread through the brain. I mean, this is something that actually I wanted to do for since I first got involved in prion research more than 25 years ago, and but you know, there was never a way to do this in a rational manner. And then Daniel Kirschenbaum, who sounds very German, but he's actually a Hungarian, and he joined my lab as an MD PhD student, and Francesca Catto, a PhD student from Italy, and they have done a couple of things. So you may be familiar with this technology called Clarity that was invented by Karl Dijsselroth, a neuroscientist from Stanford, several years ago. And Clarity is a way to extract the lipids from the brains, here from the brains of mice, but you can also do it from humans. And this way, because the lipids are actually what makes, what prevents the brains from being transparent. The reason is that is actually refraction, because the moment the lipid membranes constitute have a different refraction index as the aqueous solution, and that's where light gets scattered, and because it gets randomly scattered, it will not go through the tissue. So the idea was that if you take out the lipid and render the refraction index homogeneous within the brain, you may be able to make it transparent. And that works actually very well. But, well, I should say it works very well, except that it doesn't work very well. and the reason is that, and you can already imagine here what the reason is. The reason is that if you put the brain in a buffer, the resistance of the brain is much higher than that of the buffer, which will contain an ionic solution, and therefore the electrons will not go through the brain. And so Daniel had this intuition, and he, actually it was, you know, with hindsight you could argue this is trivial. It's the egg of Columbus, but nobody thought about this before. So what he essentially did was to create an insulating layer with mineral oil that would separate the anode from the cathode, such that now the flow of electrons would be forced to go through the brain. And this is what we call the crystal technology, and essentially it works incredibly well. You can, so for mouse brain, you can clarify them. You see here we have millimeter paper that is put underneath the brain, and you see that the brain has become largely transparent within a couple of hours. It goes very, very fast. And in fact, we have played around with human material, human biopsies. You can, small needle biopsies, stereotaxic biopsies, for example, you can clarify them within 10 minutes. So it's very, very powerful. And Now, however, clarifying tissue is one thing, but then you still have to state it, because if it is simply transparent, there is nothing you can do about it. So the thing that Daniel has invented is a second technology, which is called HITS. And this is, again, an electrophoretic technology. And here you actually place the brain in an electric field, and you have a dye. The dye must be charged, because otherwise it will not move by electrophoresis. And the dye is cast in low melting agarose, so that you can actually use small volumes. That's important, because most of the dyes are actually quite expensive. So, and then you can actually pull the dye in the brain through the electric field. And for that, well, what I should say is that, so what kind of dyes can you use? Well, it turns out we tried with antibodies, and I tell you that antibodies don't work, probably because the porosity of the tissue, the way this works is that, so you extract the lipids, and then you imbibe the tissue with a hydrogel that is mostly consisting of polyacrylamide. And the polyacrylamide, just like your STS-based gels, has a certain porosity, and of course, large molecules will have trouble going through it. And the thing is, well, antibodies are not very. I mean, we are working on technologies that will enable us to use antibodies, and we're making nice strides towards that, but it doesn't yet quite work. And so antibodies, of course, would be great, but as I said, I mean, it's difficult to get the antibody into the brain. Now, so the best dyes are small dyes, low molecular weight, and highly charged, because the charge to mass ratio is really what is going to determine whether this whole thing works. And for that, the polythiophene sounded like a great way to do this, because as you well know, I guess everybody in Linköping knows how these compounds work. You have this conjugated system of double and single bonds and the thiophene rings, and but the nice thing is that you can derivatize them. So you can put side chains, and if you put ionic side chains onto this, now you have compounds. I mean, a pentathiophene or a heptathiophene will have just a couple of hundred Dalton molecular weight, but it can carry 5 charges or 7 charges, and this makes it highly ionic. And these molecules are very strongly fluorescent, and as I'm sure you remember, the geometry of the thiophene determines the spectrum of the fluorescence, so you can create many different colors just simply by twisting the polythiophenes. And that was really, this is a slide from almost 15 years ago, and that's really the work that Peter Nilsson did when he was in my lab. He imagined that the thiophene would intercalate with prions, because if you think of amyloids, and prions are amyloids, but also A beta is an amyloid, the fold, the distance between the beta sheets is around it's around 10 to 12 angstrom, and the thiophenes are around 5 angstrom from each other. So the idea was that 2 thiophenes could actually intercalate within one sheet of prions, so one beta sheet. And the hope was that the interaction of the thiophene with the amyloid would lock the thiophene in a specific conformation. And at that time, nobody really knew what is the physical substrate for prion strains. It was clear that prion can exist in many different strains. I call them strain A and strain B. And these strains will have different biological properties. Some strains strains will create disease very quickly, other will be slower, and but really nobody knew what is it that creates these strains, and so our hypothesis here was that maybe it's a quaternary structure, the supramolecular arrangement of the molecules that creates the substrate for the strain. And if that is true, maybe actually you can report different conformations by looking at different thiophils. And okay, I will skip this, but I will just tell you that this was exactly what happened. And then later on, Uli Hermann was another MD PhD student in my lab. And Uli then, together with Beat Meyer, made a beautiful piece of work, was published in Science Translational Medicine, that where he actually resolved the structure of a polythiophene complex with a yeast prion. This is the het S, it's a prion of yeast, and essentially het S is a solenoid. So you see actually, so essentially the stacks. So here would be a molecule of het S, and here would be the second molecule, and here is the third molecule. So a solenoid is essentially a stack of molecules that are slightly rotated against each other. And here you see actually, this is NMR structure. So this is not a model, this is real structural data that from the solid state NMR that show actually what is going on. So this thiophene, which is a heptatiophene, so you see 7 thiophene rings here, and here these are carboxy residues, and you see that what happens is that the thiophenes, they go into this hydrophobic groove into het S, and then each one of the carboxy residues interact with lysines on the surface of het S. And these interactions are very weak electrostatic interactions, but you know, there is a lot of them. In this case, there is 4 of them. And this, of course, creates cooperativity. So now you have a very strong binding, because the total binding is essentially the product of the affinity of each of the individual sites. And in fact, the binding is so strong that it is essentially completely irreversible. It's impossible to get the thiophene out. Well, at least it's impossible physically, but you can actually calculate the binding energy. And this was done by Amedeo Kaflish, who is a molecular dynamics specialist, and Amedeo actually had his computer cluster calculating what he calls the unbinding energy. That is, what he does is here. What you see here is are the beta sheets of the amyloid scaffold. Then he lets in silico, he lets the polythiophene interact with the scaffold, and then he asked how much energy do you have to deliver to the system in order to pull out the polythiophene, and then this gives you a number. And these in silico experiments, I mean, this looks like a really bad YouTube movie, but it takes a supercomputer to do these kind of things, and really weeks of calculations, but it can be done with a reasonably large number of compounds, and then you can do this is loosely what people call in silico docking, and this was done for a bunch of different polythiophene derivatives, and then for each of one, then we took the most promising ones from the in silico data, and then Peter Nilsson synthesized them. By that time he was back in Linköping as a professor of chemistry, and so he made all these thiophenes, and then we put them into mice, and then it turned out that some of these thiophenes do actually nothing. So we're looking here at survival, variation in survival, and some of them are completely ineffective, but other actually are pretty powerful. And this allowed us actually to generate eventually polythiophene that are incredibly powerful. They are really, really good at blocking prion replication. And so that is a part of the story, but that's not what I want to talk about today, because what we have then done was to use the polythiophenes to stain protein aggregates in brains in whole brains. And for that, we have created little machines. This is one such machine. It's 3D printable, and in fact, we have put the plans on GrabCAD. So if you want to print it out at your place, you can. You're welcome to do that. And essentially, this little machine allows you to use the polythiophene or whatever small molecular dye you want to actually stain clarified tissue. And then what do you do? I mean, once the tissue is stained, then you want to record the image. And the way we do this is by using a machine that is called a light sheet microscope or a selective plane illumination microscope. And the way this works is that you have your sample here, and this is the objective. And now the light comes from the side. So the light comes orthogonally to the objective, and here there is an optical pathway that creates a sheet. So essentially, what you are illuminating here is a sheet that will create an optical section through the samples, which you can record. And then what you do is you move the sample using a very precise stage, and then you take another image at, for example, at 2 microns distance. Then you take another image, and so it's like tomography. So you get a stack of images, and eventually you can illuminate the entire brain. So you can do similar things with confocal microscopy, but not in this size. And we call it a mesoscope because with confocal microscopy it can only record very small volumes, but here we're talking about whole brains. So when we started this, there was no selective plane illumination microscope available commercially, so we made our own, and together with Fridtjof Helmchen and Fabian Vogt, and in fact, it's published in Nature Methods. So you can look it up, and also this paper contains the entire shopping list of what you need to actually make this thing. Here you see how this thing looks like. So here essentially is the detection optics. Here behind is a camera. Here is actually where the light comes in. So the light sheet is produced along this tract. It comes actually from both sides. So you have 2 excitation arms here and here, and this is the detection arm. And so the guys have put also some helpful labels, so you know what it is. For example, this is a chair, just in case, and so on and so forth. And it's even not particularly expensive. The most expensive part are the lasers. You see here the movement of the stage. So this is how you actually move the sample, and here the cuvette contains a fluid that matches the same refraction index as the brain. And then we bought a bunch of lasers. So here we're talking 405 nanometers, and 488 nanometers, and 594 nanometers. So we like colors, so we bought a lot of lasers of different colors. And the guys they also made a little movie because when they built the microscope, they put in a time-lapse camera on it. So again, if you want to do this at your home, you can use this video to figure out which parts go where, and then the whole thing actually was up and running. And then the question is, what do you do with it? No, and I think that what you do are spectacular things. Now, for example, here, what the guys did was to use a transgenic mouse that expresses green fluorescent protein in endothelia. So it labels essentially the entire vascularization of the brain plus the plaques. The plaques are now labeled with a polythiophene, and you see them in yellow, and in red is all the vasculature. And then you can do crazy stuff. No? So I think this is the kind of things. So here again we have a mouse that is a cross of an A-beta overexpressing mouse. So it's an Alzheimer model. It contains a lot of plaques, and at the same time it expresses green fluorescent protein on the endothelial cells. So what you see here is you see the plaques, and and once you have this data set, this is just a low resolution representation, but in reality we have this at 3 micron isotropic resolution, and this allows us to do crazy stuff. So for example, now you have all the plaques, and here is a plaque, for example, and you have the vessels. So you can start asking questions. Okay, what is the average distance between each plaque and each vessel, and is it equal across the entire brain? Does it change depending on the brain areas? So you can really run very high dimensionality statistics on entire brains. And then the question is, of course, what is the physiological relevance? And we thought, okay, the first question that we want to ask is whether the treatments of Alzheimer's disease may actually be specific to certain regions. And the way we have done this was to take a bunch of mice and to treat them actually with 3 different compounds. Ones are BACE inhibitors. This is an inhibitor of the beta secretase, so it will prevent the production of A-beta. Or with anti-APP antibodies, which enhance the clearance of A-beta, or with an actual polythiophene, because we had this idea that the polythiophene may actually work also. I mean, I showed you they work very strongly against prions, but maybe they work also against Alzheimer's. In fact, Matthias Jucker in Tübingen had already tried with an early version of a polythiophene made by Peter, and that didn't work at all. But in the meantime, Peter has better second generation polythiophene. So I thought, okay, maybe we should actually try this again. And so the mice were treated for 3 months with each one of these modalities. Then the brains were clarified. Then the plaques were acquired using the selective plane illumination microscopy, and then each brain was registered to the Allen Brain Atlas, meaning that essentially registering means that the brain is morphed and into a standard representation of a mouse brain. And this is very important because now you have the coordinates of each voxel. In the mice, each brain is a little bit different. Sometimes the brain are a bit deformed. Sometimes they may be damaged. But once you have registered them, now you have a standardized way of looking at it. So for each voxel will correspond to the same area in many different brains. And this, of course, necessitates a lot of computing. But once you have sorted it out, then you can do really great stuff. For example, here what we did was to look at plaques in mice, in A-beta overexpressing mice that have not been treated. And then we treat them with something that could be, for example, the beta secretase inhibitor. And now, because everything is registered, we can do statistics on a voxel by voxel basis, meaning that for each voxel we can ask, was the treatment successful? Were we able to remove the plaques? And now from that, we can create a 3-dimensional heat map of the effectiveness of the treatment. And this is very important. What I'm showing here are not the plaques. These are not the plaques. Is actually a very abstract representation of the efficaciousness of the plaque removal. And this you can do voxel by voxel. Then you can do the same thing for a second treatment. And then you can compare the two. And you can ask, are these 2 treatments actually effective in the same regions? or do they hit completely different regions? So this was done, and and the results are amazing. I mean, I have to say, I mean, this completely blew my socks off. I was not expecting this. So this analysis is derived from more than 100 terabytes of data. So I have to tell you that this is not trivial. Even in terms of computing power, it's at the limit of what you can do with today's technology. And but the results are amazing. So what we see is that if you compare now, and again, we are not looking at plaques here. We are looking at a heat map of the efficacy of the treatment. And what you see is that the BACE inhibitor is very highly active in the basolateral part of the brain, in the cortex, but the polythiophene in a completely different region. In fact, it's almost the opposite. It almost looks like the polythiophene is active in exactly those places where BACE is not active, and vice versa, which to me was a complete surprise. Now, so I would say that this shows you that, so the discovery here is that anti-A beta treatment are highly regiospecific, and that has never been appreciated before. And the reason is because there was never before a way to actually look at the entire brain in 3D. And but then you can even go further, no? Because now you can do spatial transcriptomics, and now the fun really begins. And because what you can do is you can go to the Allen Brain Atlas, and the Allen Brain Atlas contains, among many other interesting things, a in situ hybridization map of every single transcript of the brain. So what these people did was to make a lot of sections of mouse brains and essentially hybridize them for thousands of genes. So now the Allen Brain Atlas is a repository of the local transcriptomics of pretty much every gene that is expressed in the brain. So now what you can do is, now you have the map of efficacy of an anti-A beta treatment in the brain, voxel by voxel, and register to the Allen Brain Atlas. Now you can ask, okay, how does this correlate to the genes that are expressed in the brain? And you can do this in an unbiased manner. Just interrogate the entire transcribed genome of the brain on a local voxel by voxel basis. And you can ask which are the genes that correlate to the efficaciousness of the treatment. And this was done here, and the results are amazing. Yeah, so what you see here is again, it's a very abstract statistical representation. Essentially, what we did is to treat with BACE inhibitor, and then here what we did, what we are looking at is the likelihood of superimposition of the treatment of the voxels with high efficacies of treatment against the expression of the entire genome. And we are looking at all the genes here. And what we see is that one gene that stands out is Taiwan. Now, why is Taiwan so Taiwan has a very, very high superimposition with the efficacy of this treatment. Why is that? Well, I tell you, it's a positive control, a very good one, because in this particular transgenic mouse, the A beta, the plaque originating peptide, was driven transcriptionally by the Taiwan promoter. So it's clear that if you treat the mice, and that the removal of plaques will happen where the plaques are, and the plaques are where the Taiwan is expressed. So no surprise here, it's exactly what you expect, but it's very rewarding to see that you can identify the promoter driving the transgene simply by looking at where the plaque removal occurs. Second gene that comes up with a very high superimposition is BACE1. Well, BACE1 is the beta secretase. So again, we are treating with a compound against the beta secretase, and what you expect is that the compound should actually work in those places where the beta secretase is active. So it's not a surprise that there is superimposition opposition between the efficaciousness, the efficacy of the anti-A beta and the expression of the enzyme which constitutes the target of this molecule. So all of this is nice and good, but then you have other things. So you have CXCL1, CX3CL1, all of a sudden it's very highly expressed. And then we did the whole thing for the entire genome. And here now we are looking at a representation of of the entire genome. So we're now looking at more than 20,000 genes. And here, again, here we have base, here we have CX3CL1, here's Cy1, but then we have actually genes that are much better superimposable than even our positive controls. And what we are doing here is to compare the MI, the affinity, the superimposition measurement for the beta secretase inhibitor against the lin 5044, the polythiophene, and lin stands for Linköping, by the way. And now it's very surprising, no? Because what we see is that the base inhibitors are particularly effective in or are superimposable to genes expressed by oligodendrocytes. So here we have myelin basic protein, proteolipid proteins. These are all oligodendrocytic genes, whereas the lin 5044 is mainly active or is mainly superimposable with neuronal genes, neuregulin, SNAP25, CAM2 kinase. And so this gives you a glimpse of where this whole thing is going. I think I'm very, very, very excited by all this. Right, and now we can go to humans, and this is the work of Anna Maria Royce, who is an MD PhD student in my lab as well. I mostly have MD PhD in my lab, and she has been taking human hippocampi. And this is one so here we are looking at A beta plaques stained again with the polythiophenes, and here she is doing this is actually antibody stains, which works in this case, but it's a slightly different technology, a technology called iDisco, and arteries, and here she's interested in the vascularization of the hippocampus in Alzheimer's patients. So here you see the plaques, and here you see all the vessels. And again, then you can play the same game. No? So this is now a human brain, and we are looking at the plaques, at Alzheimer's plaques, a patient with Alzheimer's disease. We are looking at the plaques, and we are looking at the vessels. So that's where the whole thing is going. And in fact, and now we kind of grew wild, and we are doing a lot of, studying a lot of different conditions using these technologies. And we have started with A beta because I thought that this would be a low-hanging fruit, that in a way that A beta would be easier to detect than prions, and in fact that turned out to be the case. But by now we can actually find prions as well. So this is really moving very, very nicely. And then we started to collaborate with Petros Koumoutsakos, who is a specialist he's a computer scientist, and he's a specialist in simulations, flow simulations, and the idea is to simulate the entire blood flow in a human hippocampus and also in a mouse brain. And for that, you need extremely precise measurements. You really need to have all the vessels, and they need to be connected to each other. And then you can actually do these things. And I tell you that Petros is, I think he's the greatest computer scientist I ever met. He's a very famous.
That it should be benefit other students in neuroscience, obviously, somehow, and coordinate with that. And
excuse me, someone's speaker's on here.
Seriously, during the first year, my first priorities to those are.
And yet, thank you very much. Sorry for that interruption, Adriano. Go ahead.
No problem. This happens all the time with Zoom. And but anyway, what I wanted to tell you about this is because Petros, he's the head of computer science at the ETH in Zurich, and he came to me a couple of years ago, and he told me, he has been doing some really groundbreaking work on chaotic fluids simulation in hydroelectric turbines, and so that's what he has been doing. And he told me, look, I could continue to design hydroelectric plants until I retire, or I go into something more interesting, which could be actually neurobiology. So that's how we started a collaboration, and now we share a postdoc, Athena Economidou, who is fantastic, is really, and they are using actually our data to create this type of flow simulation. Right, so I think that I actually could go on for the entire afternoon, but I think that this is maybe a good time point to stop and to thank all my colleagues. And it's a big lab. I have only shown you a small part of what we are doing, but I thought that you might be interested, particularly because of the Linköping connection, and most of the work was carried out, as I said, by Daniel Kirschenbaum, Francesca Catto, Anna Maria Royce, and Athena Economidou. And I wish also to thank all the funders, and particularly the ERC and the Nomis Foundation, who has been really instrumental in allowing me to do this thing, and also an amazing donation by Dr. Hans Salvesberg, which very, very generous donation, which has allowed me to do things that I would never be able to do using the usual type of competitive funding. And so because then I found myself with a bunch of money, I could do whatever I wanted, and I could do things that were simply too risky to be written in a grant. So that was a really game changer. Thank you very much.
Thank you very much, Adriano. That was a fantastic speech. And of course,